Please, I try to be here. Can you see me from here? I'm a little bit small. I'm actually talking very fast, a set of 3 studies that eventually led us to learn quite a bit about cellulose. And cellulose is a very interesting thing because it's made just of simple glucose molecules, and you can't really understand that it comes out being one of the ancient metamaterials that were ever done. It looks like something very, very simple, but it isn't. I've been studying that with X-ray photoelectron spectroscopy. And once again, you see, that's the old Einstein way of saying that you irradiate material and it kicks off electrons from atoms. You took a look on how fast these electrons are going, and you end up knowing where it came from. So you have chemical analysis. The big thing of XPS is that it's looking at the topmost 5-10 nanometers only. So it's fantastically surface sensitive. But the thing is, since we are trying to see moving electrons, we need ultra-high vacuum, which is very, very, very dry environment. Whereas cellulose, it's abundant, it's cheap, it's sustainable, it's fantastic, it has been used forever. Especially the new nanocelluloses might replace eventually parts of glass, plastics, and steel if we can control the surface properties. So looking at surface looks good. But cellulose is very, very fond of water. So does it work out? When I came to this university, Otaniemi, it was TKK then, I said, we'll never do quantitative XPS analysis on paper. Well, see, if I talk of the good part, that is actually on paper. So let's look. We can monitor paper-making processes. This we have been able to do for a while. We can either take a look on atomic concentrations of oxygen and carbon, or we can look at the specific cellulose signal, because we have 2 different carbons in there, and we see a cellulose signature in carbon plus the non-cellulose part. And you put the atomic concentration in one place, you put the non-cellulose carbon on the other place, you place everything that's in the wood, whether that would be cellulose, lignin, or extractives on the same graph, and all of a sudden you have a quantitative method of looking how you could refine the thing. Not only that, in a good part, actually, we learned out that cellulose is the ultimate XPS in-situ reference, and this is for the physicists. It stays clean and stable in its paperback for years. It's stable as you can't really imagine what else it is. So you can do reproducible data just by taking that and putting it to ultra-high vacuum and measuring it. However, if there's something going in the vacuum, you see that too, because it actually catches up everything that is in the vacuum, including outgassing. So it's stable in air, it's reactive in ultra-high vacuum. Looks like an ideal reference material, but what about the stability? This is the big thing, and this was the good part of the cellulose. If we go into the hard or complicated parts of the cellulose, for the last 6 or 7 years, we have been actually studying nanocelluloses, and they just didn't behave for the first 4 years. How come? When the original normal paper is put into this graph that I just showed you, it looks nice and tidy there forever. In nanocelluloses, there's no tidiness and no predictability. It's just all over the place. So we were asking, is that a matter of drying? And in order to do that, we dried the cellulose, nanocellulose, and it was actually a gel with 2% of cellulose and 98% of water. So this is how much it actually likes water. Solvent exchanged it either to toluene or to ethyl acetamide, or just right in air as we do with normal celluloses. And then we go back and look how the carbon atoms should look like. After drying, see the first one up there. This is the in-situ reference paper that we use always, and it looks as cellulose should look like. As for the others, you get a fantastic big thing of something on the other 2. Only in the D-methyl acetamide dried or solvent exchange NFC gave really good and nice spectra. And this was like first time ever, so that was published in Soft Matter last year. But we didn't really explain what was going on then. We did try first. It could have been just an artifact of the vacuum, but it looked also at AFM with these cellulose fibrils. And the DMA, dried NFC, it has the nice fibrillar structure, while the toluene one did not. Actually, it looks really bad in a bottle, while this is well dispersed. And if we add silicon, try to cellulate the surface, we do get nice cellulated surface on the DMA nanocellulose, but we didn't get that into the toluene one. We had XPS results that I don't have time to show, but it was showing that this actually is the reaction we were looking for. So these CC levels actually do correlate with NFC reactivity. The question is how. I have another bad story, but I'll leave that over because I don't think I have time. There's carboxylic groups that are behaving very funny in celluloses altogether. But we go to the last one, and this is the wood. And I'm going to tell you two very short stories. One is the forever wood, and one is never the same wood. In the forever wood, we were looking at the wood aging. And we had like logs from Switzerland, 1 year, 200-year, and 500-year-old logs, which were cut and extracted and unextracted, and then looked how it looks in XPS. You know what? They looked all the same. We extracted them and had the second look. And once again, they looked all the same. So the wood inside composition does not seem to change much in 500 years. However, when we are dealing with very fresh wood, it becomes quite different. And this is the never the same wood. This time we used fresh pine directly from the freezer. And we dried it via 4 different routes, the way we did with nanocelluloses, sort of, except that we dried them a little bit different way. And once again, we are looking at carbon, one as high-res data. It's not only cellulose, it also has other wood constituents. But it should not have looked like this. With a fresh pine, it's the first experiment, it's the second experiment, it's the third experiment. Within 5 minutes, with the same material, and that's how it looks like. So this is unstable. Heat-dried one is stable, but too high. Wet extracted pine, this is what looked like the nanocellulose. And then we have heat-treated, and that's all something. So drying route matters in here. Putting a summary here, we have the stable and good paper, we have the bad nanocellulose, and we have the wood that needs stabilization. And finally, we started seeing what's going on with this nature-derived cellulose, which is structure upon structure upon structure. You look at very fast on how the fibers is going, and you have the cellulose molecule, which is self-assembled into nanofibril, which is self-assembled in the microfibril, which finally makes the 30-micron wood fiber. And the interesting thing is this nanofibril, because this is how it is synthesized in the wood. You have these rosettes that make up to 36 chains of cellulose. And at once they got out from the cell wall, they self-assemble. And they self-assemble in a very, very specific way. These are 2 chains of cellulose. On the inside, all the hydrogen bondable places are so tight there's no place for water. On the outside, you have all these hydroxyl groups that would otherwise could hydrogen bond, and they love being in water. So you have water-friendly outside, photo-free inside. You look at this cross-section, and you see that all these chains are lined up with a hydrophilic phase and hydrophobic phase coming out on the cellulose surface. And this is finally the secret success of a cellulose nanofibril. It has a water-free inside, and it has very, very specific 2 regions outside in this 5 times 200 nanometer little structure. And the big thing is when you come from wet to dry, you have to do something with all these hydroxyl groups, because when you take the water out, these are the ones that become active. And this is the one that's changing the whole image, what's going on. So it's all about surface free energy minimization. We can do it now, but it's actually nanocellulose just about all surface, so you have to know how to make on that. Conclusions, this is the final draft. Cellulose adapts to the surrounding medium. Acetone and DMA assist in self-stabilization of the surface hydroxyls. And life has evolved for 500 million years. That was the Cambrian explosion. If today cellulose reigns exclusively in plant kingdom, there has to be a reason for it. It's not magical, but it's ingenious. Thank you for your attention.
questions or comments? Not too. Okay, yes, we have one.
For a very lively presentation, being a water person myself, of course, also nice to hear on different scale. Just to try to get a proper grip on things, so what kind of practical kind of applications you see for the research you've been carrying out?
Okay. Nanocelluloses are the kind of thing that once you can do the surface right, you can make material like films that are transparent like glass, bendable like plastic, and almost as hard as steel. So this is a sustainable and green material, but you have to get along with the forces and to get the water out in order to be able to work with that. It's 2-3 big projects. We have 2 first plants in Finland going on that, so it's at its start now.
Okay, thank you.